Hi, everyone. I am going to go over Lab 05, which is the chemistry of life, the experiment one on testing for proteins. I am going to start with, well, I want to say this first. This is my fourth time recording this video, so hopefully fourth time is a charm. But the reason it's my fourth time is because and this is one of those labs that you really need to prepare for it before you do it, and that includes instructors. So I have learned a few things in my first three attempts to do a good job with this with this video. I've learned a few things, and one of those is to get your materials together, read your procedure, watch this demo first. Okay, do all that first before you try to actually do the lab. For this particular lab, you're also going to need to get some materials ahead of time. You're going to need to get an egg. You will need an egg out of your refrigerator. You will need to get some water at room temperature. So just get it out of the faucet and let it sit for, maybe 30 minutes. And in fact, you can do that while you are boiling another container of water. So you can do that on your stove or in your microwave. But the main thing is, I know you can't see this. Let me move it over. I'm using a hot plate. Of course, you'll be using probably your stove or your microwave. But my water is not boiling yet. I hope it will be, but it's more important that you have boiling water. But I have a beaker. Beakers pour easily. You could put on, you could use not gloves, but oh gosh, I can't think of the word, but you know what I mean. You can use a cloth or something to help you to so you won't get burned on the pan or whatever you're using to boil your water. But mine's not that hot yet. So anyway, make sure that you don't get burned. And if you need to pour that water into a pitcher, like a tea pitcher, something that has a spout on the end. I'm using a beaker, but you may not have a beaker at home. And you probably shouldn't use the beakers in the lab kit because you may need them for something else. So anyway, you're going to make sure you go ahead and boil your water. The boiling water is very important. So boiling water, room temperature water, egg, that all comes from you. And then you're going to need two 250 milliliter beakers. You're going to need Biuret solution, which is a blue solution. You have 2 bottles of this, but you're only going to need 1 for this lab. You're going to need an unknown solution. It's called unknown solution. You'll use the entire bottle of that unknown solution in this lab. You'll need glucose solution, and you will not use the entire bottle. You will need 5 test tubes that you have labeled 1 through 5. I don't know if you can see this, with your marker, 1, 2, 3, 4, 5. I've written directly on the test tubes for this. You will need a 100 milliliter and a 10 milliliter graduated cylinder for measuring, and you will need 5 of these. You need 5 pipettes. And it's very important in this lab for these pipettes that you don't use the same pipette in different solutions. So don't use one pipette to get your Biuret. Sorry, I'm knocking everything over. Don't use one pipette to do your egg white solution and that same pipette to get your Biuret solution out with. You don't want to mix them. That's why you're using 5 different pipettes, because you're going to use one pipette per solution and not mix things up. And you can actually throw the pipette away when you're done with it, and that way you won't make that mistake. Okay, another thing I want to tell you is this is the first time we're using the glass test tubes, and they come in bubble wrap. I would suggest, with this being one of the first few labs that we're doing, that you put them back in the bubble wrap, wash them out, let them dry, put them in the bubble wrap, just in case we need them again. I don't really think we do, but I mean, just in case, that's just a little, I'm going to keep mine, of course, but anyway, another thing that you may want to do is when you go to the lab, when you go to Lab 5 on Moodle, when you click on it, to the right, there is the lab workbook, and the lab workbook contains your tables that you fill, well, it's just one table, but it contains the table that you fill in, and it contains some pre-lab questions, and it also contains a place for you to write your hypothesis about what color each tube is going to turn. And I'm going to suggest that you do the hypothesis before you start the lab. So I'm going to give away a little bit of this. Every solution at the beginning is going to be clear or cloudy clear. You know, it's going to be some shade of clear. So your hypothesis is going to be whether or not there's protein in that solution. If there is protein in that solution, this blue Biuret solution will turn either pink or some shade of purple. So go ahead and hypothesize for test tubes 1, 2, 3, 4, and 5, what color you think they will turn. Either they will remain blue or they will turn some shade of purple because they have protein in them. Go ahead and do that because you don't want to stop in the middle. And the reason is because one of your solutions is made with Jello or gelatin, the gelatin part of Jello. And if it hardens, you don't have a solution anymore. You have a gel. And you can't mix the Biuret with the hard Jello. You can only mix it with the Jello before it hardens. So, little bit of a something that I learned in my first 3 times. Okay, so you're going to look at your procedure, and your procedure, you're going to start with test tube number 1, which is the albumin. All right, we've labeled our test tubes. Let me show you where you get albumin from. You get albumin from, in fact, I'm going to do this right into this 250 milliliter beaker. You've got your egg, and you're going to separate the egg white out from the yolk. And the way that you do that is you crack the egg. But whenever you take one half off, you leave the yolk in the other half. And then you just kind of go back and forth with it and let the white fall into the beaker. And then you're going to discard the yolk and the egg unless you want to use it for some cooking recipe that you just need egg yolk for. Remember to wash your hands with soap. I'll do that later, but I did wash them a little bit. All right, so now you're going to add 25 milliliters of room temperature water. And I'm going to do this fast. Remember when you do it that you want to get down on eye level and make sure that you're reading it exactly 25 with the graduated cylinder. So I've got my 25 milliliters of room temperature water. Then I'm going to take a pipette and just kind of mix it up. Mix the water in with the egg white. The egg white contains something called albumin. Albumin is also found in human blood. Helps to thicken the blood. And what we're going to do is you're going to try, this is a thick solution. So you're going to try and squeeze the bulb all the way and draw up 5 milliliters. And 5 milliliters in the pipette is the top mark. So good luck with it. It's not easy. My bulb is not even, and my pipette is not even, it's too thick to go into the pipette. So, all right. Now, I was patient and actually got my 5 milliliters. So you're going to put your 5 milliliters of albumin solution, egg white solution, into test tube 1. And then discard that pipette and set your 250 milliliter beaker to the side. Next thing you're going to do is you're going to make a solution in your second 250 milliliter beaker. You're going to make a solution of gelatin and boiling water. Now mine is hot, it's not boiling, but I think I can still do it. Your gelatin packet, it's a brown packet that comes in a clear packet. You're going to take the gelatin out of the packet. I'm just going to have to pretend because I've already used mine up. But anyway, you're going to pour your gelatin into your 250 milliliter beaker. You can throw the packet away. And then you're going to add 50 milliliters of hot water. So again, be very careful Make sure that you have some protection so that you don't get burned. Pour the boiling water into something with a spout on the end so that you can easily pour it. And then you are going to measure out 50 milliliters of that water. Get down on eye level and make sure it's actually 50. And then pour it into the 250 milliliter beaker with your gelatin. Use your pipette to mix it up. Mix, mix, mix. And then you're going to take 5 milliliters of that and put it in test tube 2. All right. And then throw that pipette away. All right, the next thing you're going to do, the next solution is the glucose solution. And you're going to get 5 milliliters of the glucose solution and put that in test tube 3. And then I showed you how to do it, so I'm not going to do that. And then test tube 4, you're going to measure out 5 milliliters of your room temperature water. You can do that with your 10 milliliter graduated cylinder, just half of it. Go up to the 5. And then that goes in test tube 4. This is your negative control. That means it shows you what a negative test looks like. A negative test means the solution does not have protein in it. So we know water doesn't, and so water is our negative test. This is what a negative test looks like, so you can compare it to the positive test. Then in test tube 5, your unknown solution, you can take the whole entire bottle. You can actually pop that little dropper part off the end and pour that entire bottle into test tube 5. It's kind of a thicker, cloudy solution, but all of the solutions are clear. So in your, what you're going to do now is record in your table the color of every solution in test tubes 1 through 5. And you can say anything from completely clear to kind of cloudy white, you know, just in your own words, what the color appears to be in the test tubes. Because some of the solutions are more cloudy and white and some are completely clear like water. Okay, so after you do that, you're hopefully going to have already made your hypotheses. So you've already got your hypotheses as to what color test tube 1, 2, 3, 4, and 5 are going to turn. Now you're going to take your Biuret solution. I recommend you just remove the dropper part so that you have just an open bottle, and take your pipette, and the 1 milliliter mark is the bottom mark. You're going to add 1 milliliter to every test tube. I'm just going to add to test tube 4, but you're going to do 1 milliliter in each of the 5 test tubes. And then you're going to do a very important part, which is to mix the contents of the test tubes. You'll take, and this is a technique called flicking. You'll take the test tube, hold it by the tip, and then take your finger of your other hand, your index finger, and just flick that test tube. And that will mix the contents very, very well without you spilling anything or breaking any glass or putting your thumb over the tip and getting chemical on your thumb. So anyway, after you do that, you're going to have some different colors in your test tubes. You'll record your results. You will take a nice picture of yourself with the test tubes, with the completed test tubes mixed with the Biuret solution, so that this is documentation for me that you have actually completed the lab. And then you'll upload that photograph, and you'll upload your completed lab workbook, which contains pre-lab questions, a table, and post-lab questions. And you'll upload all of that into your assignment for Lab 5 under Chapter 3 in Moodle. If you have any further questions, please feel free to email me or call me and let me know.